Hello, this is Ashwin, and today I'm going to present my research project titled CRISPR-Cas9 Targeting and Ligand Docking Study on PRNP for Fatal Familial Insomnia. So, this project uses entirely computational methods to model what a potential therapeutic strategy might look like by targeting both the gene responsible for the disease and the misfolded protein that results from it. The two main tools I'm using are CRISPR-Cas9, which allows us to edit the genome, and molecular docking, which which simulates how small drug molecules might interact with the protein involved in the disease. So these two layers of approach, one at the DNA level and other at the protein level, allowed me to get a complete picture of how a condition like FFI might be addressed in silico before moving into the lab. So now I'll be walking you through the biology of the disease, the reason for selecting the PRNP gene, the details of how guide RNAs are designed and validated, and the steps taken to model how a drug might bind to and inhibit the misfolded protein. I'll also explain each tool used, how they connect in this pipeline, the data I retrieved, and finally we will reflect on the broader implications of the findings and where the project could be extended. So Introduction to Fatal Familial Insomnia. Let's begin by understanding the disease we are dealing with, fatal familial insomnia. It's one of the most chilling and heartbreaking examples of a genetic disorder, largely because it robs a person of sleep, one of the most fundamental physiological needs, as we all know. So FFI is a rare autosomal dominant prion disease, which means it is inherited and caused by the misfolding of a protein, not by viruses or bacteria. The mutation that causes FFI is known as D17-8N, and it occurs in a gene called PRNP, short for prion protein. This gene encodes a protein called PRP, which normally resides on the surface of the neurons. In a healthy state, the protein is folded correctly and performs functions that are still being studied, possibly in cell signaling or in neural protection. But when mutated, it misfolds and turns toxic. Now, here's what makes FFA so devastating. It specifically targets the thalamus, the part of the brain that acts as a relay station and plays a key role in regulating the sleep-wake cycle. Over time, the thalamus degenerates, and you know what's the result? The patient gradually and permanently loses the ability to fall asleep. This isn't a mild problem, mild form of insomnia. It's it's a total sleep loss. So eventually, the body and brain cannot function without rest, leading to cognitive decline, uncoordinated movement, hallucinations, and finally, death. The figure on this slide contains PET scans and illustrates this perfectly. On the left, you will see a healthy brain and active thalamic regions glowing right here, indicating high metabolic activity. On the right is the brain of a FFA patient. The center is dark, indicating the thalamus is no longer metabolically active. So this single dark spot represents a total shutdown of body sleep regulation system. And understanding the genetic root and molecular mechanism of this disease is crucial if we ever hope to build effective therapies. That's where my project comes in. So the gene or the heart of the FFI, gene of F, the gene of the heart of FFI is PRNP, which encodes for the prion protein. And under normal conditions, the prion protein, also called PRP, has a well-organized alpha helical shape and is harmless. However, when the mutation like D178N occurs, the structure changes dramatically. So the protein starts to fold into a beta sheet rich form. Now, why is this dangerous? Because beta sheets are sticky and prone to aggregation. These misfolded proteins begin clumping together and form what we called amyloid aggregates. These clumps are neurotoxic and begin disrupting normal brain functions. What's even more concerning is that this misfolding is self-propagating. One misfolded prion protein can convert healthy ones around it into the same misfolding form, essentially creating a domino effect of protein destruction. That's what makes prion diseases so fast and moving and severe. Here, this figure illustrates on this slide shows the figure on the slide shows the structural change from the alpha helical PRP here to the beta sheet PRP. So the normal version is more flexible, curved, and smooth, while the misfolded form is rigid, sticky. And this change in physical structure is the fundamental biochemical event behind diseases like FFI. And understanding the switch from a benign protein to a pathogenic one gave me a very clear direction. We either need to edit the gene that produces the faulty protein or try to stabilize the protein so that it doesn't misfold. And that's the crux of this project. So the project focuses on two strategies to address fatal familial insomnia. The first one is designing a CRISPR-Cas9 guide RNA to target and potentially disrupt the mutant PRNP gene. And the second is simulating undocking of a small molecule drug. In this case, I used quinacrine to see if it can bind to the misfolded prion protein and potentially inhibit the harmful activity. In this dual approach, one genetic and one pharmacological, aims to explore how both prevention and intervention can be modeled through computational biology. My personal goal was to understand how accessible tools like benchling, IDT, gRNA designer, AlphaFold, and Twisttalk can be used together to model these solutions, even in the absence of the wet lab setup. So to better understand how this project come together, here's a step-by-step flow of the process. Retrieve the PRNP gene sequence, then annotate and validate the sequence using benchling. The third is to design guide RNAs using IDT's CRISPR tool, then retrieve the prion protein structure from AlphaFold, get the quinacrine structure from PubChem, run Swissdoc to stimulate struct protein binding, and finally to analyze binding energies and visualize results. So here I, I got the PRNP gene data from the NCBI. So after retrieving it, my next step was to validate the region where CRISPR could be applied. I used Benchling, a cloud-based platform that helps visualize and annotate DNA sequences. I uploaded the PRNP sequence and zoomed in one of the region between base pairs 469 to 621. Right here. This was from 469 to 621. And which includes the site of the D178N mutation. And that's why I chose this part of the gene. And this section was chosen not only because it spans the mutation, but also because it allows room to find a suitable PAM sequence, which is essential for Cas9 targeting. And here, it's a screenshot from Benchling. So you can see the cut sites for restriction here. Enzyme, it shows the enzymes like BST2 and BTSL. These cut sites are essential in lab setting for confirming whether a CRISPR edit actually happened. And yeah, even though this is a computational project, using these markers makes it easier eventually move to wet lab validation. In summary, I would say benchling gave me both a visual and functional confirmation that the region is ideal for gRNA design and potentially validation using standard molecular biology techniques. So once the target region was validated, I used the IDT CRISPR gRNA design tool to generate guide RNAs that target the PRNP gene near the mutation site. It, this tool analyzes several factors, most importantly, the on-target score, which tells you how well the gRNA will cut the intended DNA sequence, and the off-target score, which estimates the chance of unintended cuts elsewhere in the genome. Out of the 36 options, I selected the guide RNA with on-target score of 75, so which is relatively high and indicates high efficiency. This guide also had low predictor of target effects, making it ideal for precise targeting. A figure, yeah, here it's better. So the table on the slide, it shows the guide RNAs ranked by score. My selected guide is highlighted. You can see it aligns closely with the mutation site and has a nearby PAM sequence, which is critical for Cas9 binding. With this, we have completed the genetic part of the workflow. The sequence, it's validated and region is annotated, and a high confidence guide RNA has been selected. Moving on, now we shift focus from the genome to the protein, specifically the misfolded prion protein that causes disease in FFI. So if someone is already symptomatic, gene editing alone might not help. That's where molecular docking becomes valuable. It allows us to simulate how a small drug molecule might bind to the protein and block its activity. Um, I chose quinacrine, a compound that has shown antiprion activity in trial studies. That small crosses the blood-brain barrier and was previously used as an anti-malarial drug also. And that's the reason I chose to try. So moving on to Swiss dock setup. To run the docking simulation, I used Swiss dock, a web-based molecular docking tool. For the protein, I downloaded the structure. For the protein, I downloaded the structure of the human protein from AlphaFold, which uses AI to predict 3D structures from amino acid sequences. Then I retrieved the 3D structure of quinacrine from PubChem and converted it into a format compatible with Swissdoc. Here you can see. I converted it into a like MOL2 format. I performed what's known as blind docking, meaning I didn't specify a binding site, but allowed the software to search the whole surface of the protein to find the most energetically favorable pockets. So this is the screenshot from SwissDoc. Those docking setup, the protein is visualized, the ligand is uploaded, and the docking parameters are set. I mean, these are the default settings. I didn't change. So this is the digital equivalent of running a chemical reaction in a beaker, but instead it happens through code since it's in silico as well. So docking results. Mm, Once the docking simulation completed, the stock generated about 20 possible binding poses. Each pose has an associated Gibbs free energy value. The lower the value, the more favorable the interaction. So the best post had a value of minus 4.788 and it has highlighted it, which suggests a more moderately stable binding. Not as strong as covalent bond, but significantly enough to indicate interaction. So that's the table shows the processes ranked by the binding energy. The top pose was chosen for further analysis, and it had low energy and a reasonable RMSG, which tells us the molecule's orientation was consistent with non-stable bindings. And this value gives us a first pass confirmation that quinacrine could indeed interact meaningfully with the prion protein. Oh, here After identifying the best pose, I use Swisstock's built-in viewer to explore the 3D interaction. The quinacrine molecule nestled into a hydrophobic groove between two alpha helices and on the prion protein. These grooves are often the starting points of aggregation misfolded proteins. So your molecule binding here might help block or stabilize the process. When we look at the figure, the visualization shows quinacrine inside the binding pocket. Important interactions like hydrogen bonding here, and, and this one shows the ionic interaction here. These are the ionic interaction, and these are the hydrogen bond interactions, and they have been labeled. So seeing the interactions visually makes it much easier to interpret what the docking score means in real world terms. We can literally see the drug dock to the toxic part of the protein. Discussion. Mm, at this point, both arms of the project are complete. The genetic one and the protein one, where in genetic where we have designed a guide RNA to knock out or edit the mutated gene, and the protein one where we docked quinacrine to test its potential to neutralize the misfolded protein. Together, they offer a combined therapeutic hypothesis, say, and CRISPR could help prevent disease onset, while small molecules like quinacrine could help patients already showing symptoms. This approach mirrors what's happening in modern drug discovery, combining genetic and pharmacological tools to create precision treatments. To talk about strengths and limitation, I'll begin with strengths. So the entire study was computational and free to perform. That's a key thing. And I used well-validated tools like AlphaFold, SwissDog, and IDT CRISPR Designer. And the approach is multi-level, targeting both the gene and the protein. The final strength would be it's reproducible and could easily be extended by others. Moving on to the limitation part, the first limitation is the structure used was the wild type prion protein, not the D178N mutant. And the other limitation could be was only quinacrine was docked. I didn't compare with other candidates. And the and I'd say no molecular dynamics were used, which would show long-term interaction behavior. And another thing is the CRISPR simulation stopped at gRNA design. There was no off-target genome analysis or repair modeling. So while the project was robust as a concept, it still leaves room for deeper exploration. Looking ahead, looking ahead, there are many ways to expand the project. I would like to dock more ligands, including this and 138B, and also I'm planning to use suramin or other antiprion compounds to find better binders. And also, I like to model the D178 and mutant specifically using AlphaFold 2 or Swiss model and rerun the docking. And the other thing would be add molecular dynamics simulations to test how long the drug remains bound in the realistic dynamic condition. And finally, to simulate homology directed repair on non-homologous and joining pathways to see how CRISPR edits would play out in the real cells. So the future would work is all about converting the current limitations into a practical implication kind of thing. So basically, these steps would take the project from being a purely predictive to being part of a drug discovery pipeline. And, well, these are the references I've used for this project. Thank you.